Hi everybody, welcome to this part two of a two-part series on RNA-Seq using Jupyter. I'm Jason Williams, coming to you from Cold Spring Harbor Laboratory. Let's go ahead and get started. I'm going to share some slides, and we will recap what we were able to do last week and then go into today's activities. All right, so this is part of the DNLC Live series. And as we said, we really want your feedback, so make sure that you go ahead and visit our website at dnlc.cshl.edu and look for the DNLC Live page. And from there, you can see the previous videos, give us some comments and some feedback, and also through our various social media platforms. Of course, this video is going to be premiering on YouTube, but through any of those mechanisms, please make sure that you are liking and following and subscribing so that you see the latest content. Let us know what else you'd like to see, and also give us some feedback on what you've seen so far. This is an online course, so apologies, it's not a short video. We're going to aim for 45 minutes in part two here, just like part one. This is only a two-parter. The other thing is that these are going to have some exercises which you can also follow along. So you're going to need a Cyverse account. I'll mention that later, but it's all free. And we're going to post these slides, and there's an online lesson to follow along in detail. The course had this learning goals. We covered the first two last time about understanding a little bit of what an RNA-Seq experiment is. I'll briefly recap that. We are using the Linux command line, and although this is not a Linux tutorial, really this is a nice way to kind of get to understand some of what Linux is and what it's about, because if you are going to do some bioinformatics, it'll almost certainly be essential to learn those tools, because most of our tools in bioinformatics are written in this way. Then we're going to use a few popular tools for RNA-Seq. Last time we mentioned importing the data using the SRA toolkit, and we got just about to the part where we were trimming some data. We'll take a quick recap again at that, and then we're not really going to go all the way through to the end of an RNA-Seq experiment. We're not carrying it out in this course all the way to the end. I'll mention the other course where we did. We're really getting a look and feel. This is just sort of get your toes wet. So we'll visualize the results, but there's actually a lot of work yet to do if you really want to do a complete RNA-Seq experiment. But what you'll get in this course will really prepare you for that. As I mentioned, we're going to be using a tool through Cyverse. So if you go to cyverse.org, you can sign up for a free account. It is required. You can do everything that I'm doing, but you will need the Cyverse account, and it is free. So we're going to focus on alignment and visualization today, and let's go ahead and get ourselves grounded. What we're going to accomplish, like I said, after the review is we're going to go a little bit into alignment and also a visualization of those data. And this is also another shout out or shout back to the three-part series on RNA-Seq with DNA Subway. We're using very similar tools, this different platform. And there I go into a lot of detail about an RNA-Seq experiment. So if you really want to go to a platform, well, the platform is actually much easier to use in some ways than a Jupyter Notebook. But if you want to go into some of the details, I'm not going to cover necessarily to that level of depth in this series. Be sure and watch those. It's quite a bit of watching, but I hope you'll find it enjoyable and informative. Okay, let's recap the introduction to RNA-Seq. The RNA-Seq experiment, again, is extremely useful, popular, common in molecular biology. It's one of the key technologies that we use to really understand what's going on. And as this bullet states, if we want to understand what genes are active and under what circumstances, often that means that we have to know how they're being transcribed into messenger RNA. And, you know, if we use the analogy or, you know, an example, at least, a cell in the liver and a cell in the brain, those have the exact same instruction sets. And it's really this difference in gene expression, difference of what genes are active, that really determines what makes a cell, one cell different from another. And RNA-Seq allows us to measure that transcription, and we call all of the transcription going on the translation of DNA into messenger RNA as part of that transcriptome. Now, there are other things. Not every mRNA gets made into protein. There's other types of RNA that are really important in cellular processes, so I'm not discussing that at the moment. But the bulk of things that we're often concerned with That's what the transcriptome is, and RNA-Seq is the technique for measuring it. And we assume and make a rough assumption that the more RNA that you see, the more of that cellular process that's going on, the synthesis of a protein, some regulatory activity. And that's not a bad assumption. Not always true, but it's a really nice place to start. And we analyze these data often in comparison. So we'll often look at the gene expression in one tissue versus another, or in one state, such as cancerous versus non-cancerous, and look for key differences that are that are sort of prototypical of those differences. A tumor, an RNA-seq of a tumor, we would expect looks much different than a normal tissue, because that cancer is doing something. Oftentimes there's a dysregulation of expression, and you're producing a lot more of something than you normally would, or maybe not producing something that you normally would. So this was a convenient layout. You set up and you do your experimental design. I mention and go into the details of this in our previous video in this series, and a lot more in the other series. Then you need to get RNA from both your experimental group or groups and control group. And then that is transformed through a variety of molecular biology processes into what we call a sequencing library. In step 3, we use a sequencer to get the actual reads, which this time we convert RNA into DNA. So we're looking at DNA reads again. And then finally, because this is a bioinformatics lecture, we're spending most of our time on point 4, which is the actual analysis on our computers. So just a reminder of what our data set was, and also a reminder, a shout out to that this is part of a series of lessons that are being done by the Genomics Education Alliance. So you can check out, there's information on how to, or you can Google or go to our website on how you can see all the other lessons that we are piloting right now. This is part of one of them. In this particular example, we're looking at samples of mouse colon, and in some cases we're looking at normal tissue versus tumor, and then we're also looking at RD, which is the mice that have been raised on a regular diet, and mice that have been raised on a high-fat diet, HFD. In our case, we are focusing on even still a smaller subset of just looking at high-fat diet in the tumor cells versus high-fat diet in the control cells. And what we're seeing here on the right-hand side, if just mention again, are histological stainings. So in this case, that brown color is indicative of how much of the protein leptin, which is the protein, the enzyme that we're interested in, how much of that is present. And the darker the color, the more leptin you experience. So you could see, it looks just by eye, that the high-fat diet tumor has really, really high leptin expression compared to the amount of leptin expression that we see in mouse on a regular diet and in a normal colon histological sample. These data were obtained from the Sequence Read archive hosted by NCBI, and there's a lot of data there, and that's often where people will put their experimental data. And we're going to be able to access these lessons online. And there's a link in the description and a link on our live page to this lesson, because the lesson goes into a lot more detail than I'm going into. And it's really designed for you to be able to follow along by yourself. But I really hope that what this video does is enriches that experience and gives you a little bit more background information and guides you through sort of what's going on. Last time we also talked about that data, and we talked about you need to understand its quality. All the data is assessed, and that's sort of where we stopped off last time. So let's go ahead to the lesson and just see as far as we got last time. I am going to go ahead and start sharing that. Okay, so this is the leptin lesson. And last time we were at, if I click onto assessing data quality, we were talking about the FASTQ file format and assessing data quality. And again, there's a lot of detail in here, so I recommend you take a look at that. And then we were actually in a Jupyter Notebook. Now, if you remember, there's a link here called Launch This Lesson on Cyverse. So you needed to click that link, and when you do, assuming again that you have a Cyverse account, I'm already logged in, so you need to log in, and it would come up with a little window that really you don't have to touch anything other than just say launch analysis, and in a few minutes you'll have access to that. Now, I launched a couple of these a few minutes ago, so I want to launch the newest one, which is actually clean, and then I have the other one as a backup. Because I know I want to actually click on launch the lesson. One of the caveats to doing this in any type of live format is that some of these steps do take quite a bit of time. So the trimming step and also the step that we're going to do with Callisto and we're going to do alignment can take more than an hour to complete, even on a pretty fast computer. So when you're doing these things, you may want to give yourself some time, particularly, like I said, the trimmatic step, which I'm going to talk about today as part of the recap, and then also the Callisto step. If you do all of it, it's going to take many hours, a couple of hours to do those. Fortunately, and as I think we're going to do today, we have those data there as a backup already pre-computed, so we can take a look at them, and then one could go through on their own. So however you want to do it, but just a heads up that there are some parts of this experiment that do take significant time because we're dealing with tens of millions of reads. So this is a lot of data. These are real data sets. Okay, so let's go back to sharing the screen. Okay, if you remember from last time, everything that we're doing takes place in this platform, which is called the Jupyter Notebook. So in Cyverse, I've launched the Jupyter Notebook. Remember, when you go to launch, what you are going to get is a little notification. And if you click on that notification, it'll give you a link to your running analysis. Now, let me try desperately to keep these separate so I don't confuse myself. Okay, this is the clean one. And in there is a folder called Notebooks. And we went through Notebook 0, and I'm going to start this time with Notebook 1. And I'm just going to go through some parts of it just to get us up to kind of where we were at. Now, if you notice, every notebook starts off by reminding you that we are looking at these particular samples from the Sequence Read Archive, and that we've got 6 of them, all of them on the high-fat diet, but there are 3 replicates. So we do this experiment. We sample 3 different sets of mice, or probably 3 individual mice in this case, and that's because there's biological variability. And look to the other RNA-Seq lesson that I gave because it covers that very well. Now, if we go ahead, if you remember, each one of these sort of cells with a command line command in them, if you hit Control-Enter, it will go ahead and run the cell and give you the same output as you would on the command line. Just to prove that, if I copy that and launch a command line terminal, you could do it this way, and you could copy that same command, and it works. So once you do things in the Jupyter Notebook, you may want to go ahead and do things at the command line. One little tip is nobody really remembers every single command. Copying and pasting is really something we do quite frequently on when we're doing bioinformatics. So don't be afraid to copy and paste, but some of them certainly you'll get a hand of and remember how to do them. So these are samples. If you notice, there's also this one called small.fastq, which is not real. It's some smaller data. So if I change into that directory with the cd command and run this command, we used a program called fastqc last time. It will go ahead and analyze that data fairly quickly, and we notice that we're going to be left with 2 files. Now on the left-hand side, if I go back to notebooks, or rather one step back up, I think it's in data. And where did we cd into? Data, raw data, FASTQ. There is my FASTQC report in HTML. For the sake of time, I'm not going to go through this report again. I did last time, and there's also some excellent resources. But if you remember, this chart and several others that are included as part of that report are a representation of the quality score. So when we showed that little chart, things that are up here in the 30s, this is when we're talking about a 99.99% or higher quality. But if you look, on average, some of our quality towards the end of our sequence is not that great, so we're going to need to do something about that. Back in Notebook 1, there are some steps that are there where we make some directories in order to place our data in a more favorable place, and we move those things around. And also, there are exercises. Pardon me. So I recommend, if you haven't done so, to follow the exercises and try your hand at writing these commands for yourself, because what you needed to accomplish is you needed to go ahead and run FastQC for each one of the real data sets. We did not do that, but you need to be able to do that. So follow the lesson, and if you have questions or problems, let me know in the comments, and hopefully get some help. What I want to focus on for just a second, if I go back here to notebooks, is now notebook 2, where we're going to use this tool called Trimmatic. And there is a link there to explain it, but as the name kind of suggests, what Trimmatic allows you to do is to trim these sequences. If you remember from our file here, maybe just like cutting the ends off of a loaf of bread, maybe you don't like the crust. Some of this data here, particularly as we get, this is the length and position of base pairs. As we get towards the end of it, it's not the highest quality on average, so we want to get rid of some of those sequences. So if we're back in Notebook 2, and we go ahead and take a look at what's in that folder, again, we see all of our FASTQ files, and this is what a command looks like for Trimmatic. So it's broken down in the notebook, but let me break it down here for you too. First of all, the first command is the name of the software, which is very, very common. Usually we start with the name of the software we want to run, called Trimmatic. The next one is a thing called SE, and that tells us that in our case, these are single-end read data. So there are cases, in fact, it's much more common now, that when you sequenced in an RNA-Seq experiment, we actually sequence both strands of the DNA, the forward and the reverse strand. And that gives us a heightened confidence that we have high quality data because we're doing 2 observations for every read. It's a little bit more expensive, but you want that data. In this case, this is single end, so we type in SE to let the program know that this is single end data. The next thing we do is we use the word input. Or rather, we give the file. We don't have, in this case, we don't have to use the word input. It's called a positional argument. So just put them in the right order. We put in a file. That's the name of the file that we want to analyze. In our case, this fake name is input.fq.gz. And then we give it a name for what we want the output to be called. In our case, we want it to be called output.fq.gz. And then the next thing are a series of operations that you want to pull onto or put into the program. You want it to do certain things step by step in order to do the trimming. Now, these are really well described if you go to this link and open the instructions there, but I at least give you an idea of what's happening. In this case, this Illumina clip function, when we do sequencing, there are some extra bits of DNA that we add onto each read called adapters, so this would get rid of them. This leading command chops off, or you could sometimes say the word lops off, the first 3 nucleotides that you chose 3 because you entered the number 3. You could enter 30 and get rid of the first 30. Trailing is, well, get rid of the last 3. Sliding window means go ahead along all of your reads. Let's say you have 100 base pair read, and take the average quality. In fact, look at every 4 and take an average. If the average of Phred score is 15 or higher, you can keep it, but if not, you can drop it. And then this last one, min length, often comes at the end, is after you've done all of these other steps, then go ahead and if what you get out after trimming it is 36 nucleotides or less, then drop it all. So that's like if you had a piece of, let's say you had an apple that had a brown spot on it. Well, if there was so many brown spots that you cut out of an apple that there's no apple left, you might even want to just get rid of the apple, right? It wasn't worth it. So these are explained pretty nicely and in more detail than even I've just done, but hopefully that helps you with the explanation. And in fact, I've given a nice English explanation of what is going on. So let's run that command on the small file. And when we do, it tells us that it is running the command, and it was quite quick. It says that this file only has 50,000 reads, and it says that using this command or using this set, in fact, I only did the sliding window, so average of every 4, and if it's 20 or below, drop it, or below 20, drop it, and then I did minimum length, 75. And it said that 77% or 38,000 of my reads survived that level of stringency, and 22% did not. So I get an idea of how much I had to trim. I can't tell you in advance exactly what are all of the right parameters, but hopefully if you have good data, you're not going to be getting rid of a large amount of it, maybe 10%, 20%, but if it's starting to be more than 20%, might be questionable data. Now, this is a sample file, so I'm not so concerned that it's a little bit more than 20. When we go ahead and look, our file comes out. It's called small trim, so it's nice to rename the output as something that indicates it has been trimmed, and we move it to an appropriate place. As we talk more about bioinformatics, it's extremely important that you keep data that you have manipulated in some way separate from what we call the raw data or the original data. Okay, if we change into that file, the next thing we want to do is we might want to go ahead and run fastqc on that again. And in fact, if we look at what the small fastq file looked before, okay, we see that it's failing a couple of these quality control metrics, and towards the end, on average, it's not the best. So let's see, now that we trimmed this, that's going to be in our RNA-Seq project, trim reads, and let's look at what it looks like. Okay, all right, now if we look, you could see that it's a difference, right? Those bad sequences are really not there anymore. There is a little notch here where some sequences get a little bit poor. Now, if you look, we did that min length, or we did that sliding window with an average of 20, so nothing is going to have lower than 20. And if we really wanted to get rid of this, we could say choose 28. And so that we could really be sure that we're working with the highest quality DNA. So that's an example of what you want to do here. Now, the exercise, which I'm not going to do, takes a while to do, is for you to go ahead and do the trim-o-matic, but do it now on all 6 of the files. So it's going to take you time. It may take several minutes in order to complete that command. And if you're really interested in the command line, there is what we call a for loop, which is explained in the lesson. So if you go back to the lesson and you look at the command line primer, this is a really common technique in programming, and it's a really excellent tool to be able to use in Shell. What this will allow you to do, pardon me, is instead of doing that trimming one by one, you can sort of do them all together. So there is some information on how to do that. We're not going to do that for the sake of time. That is left to you as an exercise. Okay, so let me close some of these notebooks, and let me talk a little bit now about Notebook 3, because we have Notebook 3 and Notebook 4 that we need to get through. All right, so Notebook 3 is this thing called Pseudo Alignment with Callisto. And before I go into this, I'm going to go back to my slides because I have just a few slides to give us a little bit of an orientation here. Okay. So one thing I wanted to mention at this point is that this is just from a paper, and this is 2016, so things have changed, although, you know, this is still quite good. There are many, many, many ways to do RNA-Seq, many different softwares, many different paths. It's not just a matter of I'm doing an RNA-Seq experiment. It's a matter of really choosing what is the appropriate RNA-Seq experiment on many, many levels, and that includes the software that we use. Now, in our case, what we're going to use, just like in my last series, is a software called Callisto. This is published a few years back, but it's still really, really good and actively developed. And it is not only fast, which, you know, some programs can be fast and they usually trade off for accuracy, but it's also really, really accurate. So we're going to go ahead and use that because fast and accurate is good. I'm not going to go in as deeply. Again, throwback, go watch the other series. But let me just give you some of the basics. Callisto does what we call pseudo alignment, in that it does an alignment to the transcriptome. And most aligners, or many RNA-Seq aligners, align to the genome. But that's not why it's called pseudo exactly. It does a pseudo alignment because in the process of alignment, rather than checking every single read or against every single letter in a transcriptome sequence, it really checks enough of those letters to be certain that that is, likely a match, and then discards the rest. So I make some analogies. I won't go into them now, but just trust me that it's a really nice way to do this because it speeds up the analysis without sacrificing much in accuracy. There are a couple of steps. That's the other reason why we do this, because some analysis software can be many, many steps and many parameters, but this one doesn't have that many. You're going to build an index. So what you're going to do is you're going to align up your transcriptome, which is the sum of all of the known transcripts, so generated from previous RNA-Seq experiments. We know, and even before RNA-Seq, what are the genes in a mouse or in a human and many other organisms. And we also have a pretty good idea, although it's never complete, of what mRNAs are in a mouse or in a human or in an Arabidopsis plant. So we take those and download them, and we build an index against that. And then we do the alignment step. So to get that, in this case, I'll take you there in a moment. We can use something called Ensemble, where it's a public database. That's human, but we'll use mouse since we're using mouse today. And this is just giving you an idea again. Here is one read that might be generated. This is fake because there's only a few nucleotides. A real read is maybe 100 or more. But our transcripts that we may get in our RNA-Seq, or rather that we know about, this read may match several of them up to a point. So when we get to this little CAA junction, it matches the magenta and the blue and the green. But then if you go to AAG, well, that only matches 2 of those colors. And if you go AAC, well, only matches the other one. So there's this nice process. It's a nice bit of math about the Brown graphs that talk about the science of that. So we're going to do that, and we're going to take a look at the results. So let's go back to this notebook. Okay, and it goes through the steps. So the first thing we need to do is, there's a link here. Make sure that opens up in a new window. I hope it does. If it doesn't, I'll get back to it. Okay, so, well, it does take you to kind of, let's share my screen again. It does take you to kind of a blunt and not pretty page, but this really is the Ensemble mouse transcriptome. So if I were to click on these and download them, that would be the data. If I just go back, well, if I just go ahead and search Ensemble mouse genome, let's click on that. It's a much prettier page, but I could find that, and from this website you would navigate. Basically where I just was at the FASTA file download. There's a whole bunch of other information here. I wish I had time. But let's go back and focus. We'll use this wget command to go ahead and, once I click there, download those data. And we're going to make a directory to place them in the right place. And we'll make another directory for this index. What I think I'm actually waiting for. Okay, that's all done. Now, this step is something called building an index. And this is one of the functions of the Callisto software. So we say Callisto index instead of Callisto align, or whatever the right command is. And what will happen there is that it will go ahead and read that transcriptome data that we downloaded with the wget command, and then it will go ahead and move things along. Now, if I go, let's look at the notebook that I kind of completed halfway. Notebook 2. Actually, it's notebook 3. If I go to that, these are the types of outputs. It's really nothing so spectacular to look at, but these are the outputs, and it takes probably about 10 minutes to do that. Then there are a number of other data sets that you need to do. So I'm not going to go ahead and get the index. I'm just going to show you what you need to do, and I'm going to show you our cheat to save some time. You need to also get what we call a GTF file. So those are the descriptions of those transcripts, like what chromosome they're on, and do they have a name, because the first file doesn't download all that information. So we make a place to save that as well, and we move those around. And then finally, we also do another command. We need to get, just because these happen to be single read files, we need to get the length of all of the chromosomes so our mapping can be done correctly. So we run this command, and what we end up getting out is here are the 19 mouse chromosomes plus X and Y, and here are the lengths. So those are just something that's needed in our case. So the exercise requires that you do the trimming, and then what you're going to do is run this command. It's also a for loop, which goes through and analyzes those data. Now, you're going to need to read through this to really understand every level of detail. I'm not going to do that with you, but I do hope you take a look, especially if you're getting used to this. Now, in Notebook 5 is our little cheat. So Notebook 5 is called backup data, and so for any step that we did not do or somehow went horribly wrong, we can skip ahead by running these cells. You have to run the very first cell in this notebook, so we're going to run the first cell just to configure it. And then what I want to actually do is get the Callisto alignments and transcriptome files and BAM files and all of those different things, so that we can just take a look and know what's going on. So I'm just going to run this one, and I'll take a moment. I think there are a number of files, but what I want to do is give you at least a hint of what these type of files look like, so that you can know. And then once we take a peek at least what they are, I'm going to show you how you visualize them. Remember, again, this series is just sort of an enrichment over the written lesson, which is very, very detailed. But if you're at that level where you're really starting to ask questions about how to use some of this stuff, we hope you explore it a little bit more. Okay, so that's busy importing. It probably will take another few moments, but already I imagine we have some useful things. So if I navigate on my left-hand side here to RNA-Seq project and Callisto and analyzed, I'm ready to see that a number of these things have downloaded. So let me choose one of our samples. And what you are going to get, you can't look at all of these files, but the one that you most want to pay attention to at the moment is this thing called abundance.tsv, tab separated. Let's try to open that. It is quite a large file, so let's make sure. Let me take a moment. Okay, because all of our files are downloaded. All right, let's see if it can open it. There we go. All right, so what we get from the this abundance is we ran Callisto. We aligned each one of our 6 files, 3 replicates from the high-fat control, normal colon tissue, and 3 from the high-fat with the tumor. And what you end up with is this several columns. And in fact, if I go back to my other slides on Callisto results, let's just show you what those columns are. You're going to get something called the target ID. So this is just the name from Ensemble, and often it's not a very pretty name, but it's a nice name that's unique from Ensemble. And then you're going to get the length in nucleotides of the transcript exon, which is only exon. And you're going to get what we call the effective length. So this is a little bit of, I don't want to say guess, but I guess it's an estimation. When we sequence, we originally often, and this is in library prep, we chop up the RNA that we get into fragments that are often 300, 400 base pairs. So we need to correct for that. And then finally, and this is the most important thing, is we get something called estimated counts. So these are the number of reads that have mapped to a transcript. So I have, again, some explanation and a little bit of analogy to give you an idea, because you need to normalize this data. You need to correct for the fact that There's certain biases and certain ways that we sample. So to help you understand that, I brought over this explanation. If you look at these two genes, gene A and gene B, which one of these is longer? So I hope by looking, you can kind of guess that gene A is longer. And in my little cartoon example, 300 base pairs for gene A and 100 base pairs for gene B. Now let's say I have some sequencing reads that I've aligned to them. So those are the reads in black. And my next question would be, which one of these has more reads? Do you count more reads over gene A, or do you count more reads over gene B? Now you can pause the video and do this, but you don't have to. But let me make you draw your attention to something, which is that if you were to count, you'd find out that there were 27 reads on gene A. Just if you're asking a question, those little thin bars are where we have introns, so those don't show up with the thick black bars of the exonic material. But anyway, so that's a single read. So anyway, there's 27 of them on gene A and only 16 on gene B. But my question still stands, which one of these has more reads? Well, what happens is that you actually need to correct for the fact that just by virtue of the fact that a gene is longer, there's going to be more reads, more opportunities for reads to map to that gene. And if you were to take a simple ratio, this is not the only way we really correct, but this is good for our cartoon analogy, then 27 over 300 base pairs is only 0.09, and with 16 reads over 100 base pair genes, it's 0.16, which is higher. So there is definitely other analysis steps that we need in order to get an accurate understanding of whether one gene is more highly expressed or not. And so these are the types of things that we are getting in our analysis. Now, as I said, we're not going to go all of the way through an in-depth analysis of these counts and of RNA-Seq here. That's more advanced than what we wanted to do, and I draw your attention to the other series. But what we can do, and what we are prepared to do today, is just take a visualization and just by eye get a little glance at what that looks like. So I'm going to close this up, and I'm going to go to Notebook 4. Let me close some of the notebooks I don't need. So in Notebook 4, we can do a visualization. So if we have imported those data, or if you do everything by yourself and run this first command, what you will see is that you end up with these 1, 2, 3, 4, 5, 6 analyzed data sets. And as a reminder what they are. There is an optional thing. You may need a copy of the mouse genome to download, but we already provide that for you. But there's the instructions on how to do that. So if we make a directory and move our needed files there, this notebook is a little bit different because this notebook, if you pay really razor sharp attention on the right-hand side, is in Python. So it's a coding language, not the command line, the Linux command line as we were using before. That's also why when you look carefully, these commands have a little exclamation mark in front of them. That's to notify the notebook that we're still running bash commands, but the next command, called import genome view, is importing a Python software that we need in order to do visualization. Now this next part of the code, you don't need to guess at. What we're doing is we're telling this Python software called Genome View that these are the data sets that we want to visualize, and we're actually saying, we're actually picking a particular location on chromosome 6, which happens to be the location of the leptin gene. So if I run this, and then run this thing called visualization, as I recall, we get a minor warning, but in a second, what you're going to see is that if we count just by eye the number of reads from the high-fat diet and the number of reads from the regular diet, visually there's going to be a difference. That is not a substitute for an actual real statistical analysis, but it just gets through some of our point, at least for today's lesson. So I'm going to let that finish. It takes a few moments. Okay, so what are we seeing? Give me a little bit of room here. So each one of these little arrows is an individual read that mapped. So here's high-fat diet mouse, and here's control. sample. And if you were to count these, I have not, but what you say there's maybe 30, 40, 50, maybe somewhere in that realm. But if we look at the high-fat diet control, same conditions, but this is different mouse. So there are more reads, and maybe here, let's say there's 150. And here's sample 3, which is very few. So on average, let's even look at the peak and say there's 100 reads, 150, 200 reads that came from high-fat control 2. There's biological variability. But if we look at the tumor sample, there's already here is probably 50 or 60 by our eye estimate before. And then we go to tumor in individual 2. This is a different 2 than the 2 above. There's quite a number of reads that are there, many, many more. I would say that's in the order of perhaps more than 1,000 or so. And if we keep going, scrolling all the way down, we've got a lot of reads that are mapping to that individual. I'm afraid of scrolling too fast lest I not find individual 3. And there's 3. Okay, so there are by eye a lot of reads. That is not enough to draw a conclusion, but it is suggestive that maybe in our high-fat diet there's some difference in gene expression. Now, how could we see this? The last thing we have time for, a little bit more detail. We can go ahead and open this UCSC Genome Browser. And this is a Genome Browser, popular one on the website that we can actually use to go ahead and look at things in a little bit more detail. So in order to do that, it says that we need to search for a gene. So let's go here. We are going to search, first of all, we want to click on mouse because we're looking at mouse data. And we want to search for LEP for leptin. And already it comes up. And if I click go, what it's going to do is take us to the area of the Genome Browser where we can find that. Now, if you look on if you search for the Genome Alliance, and we'll put a link in our description, you can find one of our lessons on Genome Browsers to see what we're looking at. It's quite small here on my computer, but what we're looking at, we are on chromosome 6 at this position, 29,060,220. And this LEP in blue here is the gene, with this long line being intronic and then these thicker ones being exons that we have. Okay, so there's a lot of detail here, much more than I can really explain in even a couple 2 hours, so let alone a few minutes of a lesson. But we can view our data in context, and I'm going to show you how. And the great thing about all of this is if you do a different experiment, different data, hopefully this is a guide that shows you how to incorporate some of that. So what it says here is that we can do this thing called add custom tracks. So we're going to go back here, and we're going to click the add custom tracks button, and let's just do one of them. All right, and it says we need to paste in some stuff. Now, as it turns out, we could have we don't really upload our BAM files directly, but you can place those BAM files on Cyverse. And if you go to learning.cyverse.org, you can see how you can export them in Genome Browser. But for the sake of clarity and time, we've just given you what you need to copy and paste. So I'm just going to do high-fat diet control 1. Let's do 2 of them. That's high-fat diet control 1, and let's do high-fat diet control. Let's do one of the tumor samples. Okay, so I think I have this right. Looks sort of like there, and then it says that we need to submit. And what it's going to do is, if I've done that correctly and there aren't any mistakes, it's going to import that data directly from Cyverse. Now, this is a copy of the data that's in our Jupyter Notebook. If we were really doing it inside, if we really want to view our own data, we would have to save that data from our Jupyter Notebook back to Cyverse, and then we could generate this type of visualization. The other thing is, while we're waiting for that, is that back in your analyzed data, go back to here and to notebooks, or rather, I'm sorry, here and to data. Once you do, actually I want to go into RNA-Seq project. Once you've made any of these analyses, you have your RNA-Seq project folder. If you right-click click on these different things, well, entire folders you can't download, but the BAM files, oops, they're being Callisto analyzed. We use a file called the BAM file in order to download these things. These are pretty big, but you can actually go ahead and click on download. And then I'll show you how to get them from Cypress before I'm done here. Let's go back and click go to go ahead and view those tracks. It'll take a moment to reload it. All right, so now we have high-fat control, high-fat tumor. It doesn't look like all that much now, but what we want to do is actually go ahead. We can do like squish, which is refreshing them, and we don't want them quite as compressed as they were before, but we can at least put them side by side. Oops, let's make this a little bigger on my screen. And we can see those reads. Actually going to zoom out and move to the right a little bit, hopefully not out of focus. And we can see those reads there, okay? In this case, just by eye, it does look like the tumor has more, I don't know, maybe quite double from one. But it's really pretty cool that by eye you can start to see this visualization. So I encourage you to play around. When you are done, if you were to go back to Cyverse Discovery Environment, as you were previously, what you will do is, if you click on Analyses, back where this was launched, you may not have paid attention, but this tab was open when you launched your thing. Your data, all of your Jupyter Notebooks will be running in one of these analyses. And if you're done, you can click on those three dots and say complete and save outputs. And then when you come back here, there'll be a link to those files so that you could export them, or you could send them to a Genome Browser. They will end by themselves after 2 days, and you won't lose your data. It'll go and automatically write it back to your Cyverse account. So with that said, also there are instructions back here on using a separate standalone tool, which is popularly called IGV, which you could also look to download and visualize these data in a different way. And it's pretty cool. So I invite you to explore it. So let's just go back and wrap this up. Really is a lot to try to take in, but go use this along with the lesson. We were able to talk about what an RNA-Seq experiment is and learn a little bit about command line in Jupyter. We saw that you can import data from papers often from using SRA Toolkit. You can use tools like FastQC and Trimmatic to evaluate the quality of your DNA sequence data, your RNA-Seq. And you can use Callisto to pseudo-align those reads and even visualize the resulting files using tools like Genome View, as we did in the notebook, or the UCSC browser. So those are some of the activities that you can do. So that's it for this. It's a lot in 2 lessons. I think you really need to go back and take a look at some of the written detailed things that are there to assist you. But it's a great introduction to really one of the most popular experiments in the molecular lab. So also don't forget to go back to our live site, download any resources that are posted here, and then also visit some of the other materials that we produced and let us know what you'd like to see more from us. So with that, it's been a pleasure. I hope this was useful and insightful, and we'll see you in the next video. Thanks.